Special features of eukaryotic genome. Special features of eukaryotic genome: larger size, introns and exons, non-coding DNA, multiple origins of replication, chromosome structure, promoters, enhancers and silencers, then telomeres, centromere, repetitive DNA sequences, retrotransposons, DNA transposons, histones, nucleosomes, epigenetic marks, chemical modifications of DNA or histones that can regulate gene expression. Then CpG islands, regions of DNA that are rich in CpG dinucleotides and are often found near the promoters of genes. Then CpG methylation, a common epigenetic modification in which a methyl group is added to the cytosine base in a CpG dinucleotide. Then heterochromatin, then euchromatin. After that, transposable elements, you know that. I have a video about this topic. Jumping genes. Alternative splicing. Let's quickly learn this topic. Transposable elements, also known as jumping genes, are sequences of DNA that can move from one location in the genome to another. They can disrupt gene function and cause mutations, but they can also contribute to genetic diversity and evolution. Transposable elements are found in all eukaryotic genomes, and some make up a significant portion of the genome. For example, in maize, transposable elements make up 85% of the genome. Then alternative splicing. Alternative splicing is a process by which exons can be selectively included or excluded from the final mRNA transcript, allowing for the production of multiple protein isoforms from a single gene. This process greatly expands the diversity of proteasome and is estimated to occur in up to 95% of human genes. For example, the gene for the protein tropomyosin can produce over 50 different isoforms from alternative splicing. And epigenetic modifications. Epigenetic modifications are changes to the DNA and histone proteins that do not involve changes to the underlying DNA sequence. These modifications can affect gene expression and are often heritable from one cell division to the other. Some common epigenetic modifications include DNA methylation, histone acetylation, and histone methylation. Epigenetic modifications are important for regulating gene expression during development and can be influenced by environmental factors. For example, exposure to high levels of stress during early development can lead to changes in DNA methylation that increase the risk of mental illness later in life. And genome rearrangements, translocations, inversions, and duplications. Gene regulation networks, multi-signaling pathways, transcription factors, and regulatory elements. Gene duplication and divergence, non-coding RNAs, chromatin remodeling, alternative polyadenylation, RNA editing, horizontal gene transfer, then genome editing. Chromosomal aberrations, transcription factors, gene regulatory networks, alternate splicing coupled nonsense mediated decay. RNA interference, non-coding RNAs, circular RNAs, alternative polyadenylation, polyploidization, gene fusion. Importance of eukaryotic genome, basis of inheritance, cellular processes are taking place here, then gene regulation, developmental processes, evolutionary process, disease process, biotechnology and conservation, personalized medicine, fundamental research, comparative genomics. Personalized genomics, omics technologies, used in omics technologies, then bioinformatics, ethical considerations are also important, interdisciplinary collaborations, public engagement, techniques used to determine structure and function of eukaryotic genome, DNA sequencing, chromosome staining, and fluorescence in situ hybridization FISH technique can be used to detect specific DNA sequences within the genome. And along with that, chromatin immunoprecipitation, then CRISPR-Cas genome editing, etc. Major functions are encoding proteins, regulating gene expression, maintaining chromosome structure. Now let's learn some other topics like junk DNA, satellite DNA, like that. So DNA is the hereditary basic unit of life as it contains all the information related to organism. The DNA is present in the form of sets which include all the different genes and it is known as the genome. The genome contains all the viral information regarding the organism. Interestingly, all animals have a large excess of DNA that does not code for the proteins used to build bodies and catalyze chemical reactions within cells. The proportion of coding versus non-coding DNA varies significantly between species. In the human genome, for example, almost all 98% of DNA is non-coding, and in bacteria, only 2% of genetic material does not code for anything. Then junk DNA is a popular yet controversial concept that states that organisms carry in their genomes DNA that has no positive impact on their fitness. Junk DNA, non-functional DNA, is a DNA sequence that has no relevant biological function. Most organisms have some junk DNA in their genomes, mostly pseudogenes and fragments of transposons and viruses. But it's possible that some organisms have substantial amounts of junk DNA. Earlier, scientists dismissed non-coding DNA as junk with no known purpose, and they called it junk DNA. However, further research has shown that all junk DNA is not completely useless. Satellite DNAs represent the most abundant fraction of repetitive sequence in genomes of almost all eukaryotic species. Long arrays of satellite DNA monomers from densely packed heterochromatic genome compartments are also span over the functionally important centromere locus. Although their function, if any, is unknown, microsatellites have proved very useful to geneticists. Many microsatellites are variable, meaning that the number of repeat units in the array is different in different members of a species. Selfish DNA refers to segments of DNA that are capable of proliferating within a genome, often at the expense of organisms' fitness. This concept was proposed by evolutionary biologist Richard Dawkins in his book The Selfish Gene, published in 1976. The concept of selfish gene challenges our traditional view of genes as mere servants of the organism. Instead, it highlights the active role of DNA plays in its own replication and survival. By recognizing the existence and impact of selfish gene elements, we gain a deeper understanding of genetic evolution, genetic conflicts, and complex interplay between genes and organisms. Then junk DNA. A region of DNA that usually consists of a repeating DNA sequence, does not code for proteins, and has no known function is called junk DNA. It is also called a parasitic DNA, which is the dispersed or randomly repeating and genetically neutral eukaryotic DNA sequence, which has no obvious biological function. It is believed to be a defective relic of normal genes. Although functionless, it can perpetuate, spread, and accumulate in the species by virtue of its rapid replication. Curiously, it does not cause any survival disadvantage to the organism containing it. Traditional view of central dogma, DNA, RNA, non-coding regions in junk DNA is also there. After splicing and other processing, forward and folded by post-translational modifications. In 1972, the late genetist Susumu Ohno-Fitness proposed the term junk DNA to describe all non-coding sections of a genome, most of which consist of repeated segments scattered randomly throughout the genome. Junk DNA refers to non-coding DNA, which, unlike coding DNA, does not provide instructions for synthesizing proteins. Scientists have found that some junk or non-coding DNA are essential to optimum cell function, particularly regulating gene activity. This DNA contain many types of regulatory elements that play key roles in determining where and when genes are turned on and off and providing sites for transcription factors to bind. While non-essential junk DNA can be removed without any adverse effects, removing essential junk DNA can result in the dire consequences for the organism. For example, non-coding DNA contains sequences that act as regulatory elements determining when and where genes are turned on and off. Such elements provide sites for specialized proteins called transcription factors to attach or bind and either activate or repress the process by which the information from genes is turned into proteins transcription. Non-coding DNA contains many types of regulatory elements such as promoters, enhancers, silencers, and insulators. Other regions of non-coding DNA provide instructions for the formation of certain kinds of RNA molecules. Examples of specialized RNA molecules produced from non-coding DNA include transfer RNA and ribosomal RNA, which help assemble protein building blocks, amino acids, into a chain that forms a protein. About 80 to 90% of human genome has been designated as junk including most sequences within introns and most intergenic DNA. Some non-coding DNA regions called introns are located within protein coding genes but are removed before a protein is made. Regulatory elements such as enhancers can be located in introns. Other non-coding regions are found between genes and are known as intergenic regions. Junk DNA may act as a protective buffer against genetic damage and harmful mutations. Non-coding DNA, also known as non-coding regions of junk DNA, refer to the segments of DNA that do not encode protein sequences. Non-coding DNA can be divided into different categories based on their functions or characteristics. Introns are non-coding segments found within genes. When a gene is transcribed to RNA, introns are removed during a process called splicing, and the remaining coding segments called exons are joined together to form the final RNA molecule. And regulatory elements. Non-coding DNA contains regulatory sequences that control gene expression. This include promoter, enhancer, silencer, and insulators. Promoters initiate the transcription of genes, while enhancers can increase gene expression levels. Silencers, on the other hand, can repress gene expression. Insulators help in organizing the three-dimensional structure of chromosomes. And transposable elements. Transposable elements, also known as jumping genes, are sequences of DNA that can move within a genome. They can replicate and insert themselves into different positions within the genome, potentially affecting gene regulation and genome structure. Transposable elements can be classified into two main types: retrotransposons, which move via an RNA intermediate, and DNA transposons, which move directly as DNA. Telomers are repetitive DNA sequences found at the ends of chromosomes. They protect the chromosome from degradation and provide stability during DNA replication. Then repetitive DNA sequences are present in multiple copies throughout the genome. They can be classified into two copies: tandem repeats, where the sequence is repeated adjacent to each other, and interspaced repeats, where the sequence is scattered throughout the genome. Pseudogenes are non-functional copies of genes that have accumulated mutations over time, rendering them unable to produce a functional protein. Next topic, satellite DNA. Satellite DNA or SATS DNA is a highly repetitive DNA consisting of short sequences repeated a large number of times. It carries a variable AT-rich repeat unit that often forms arrays up to 100 MB. The monomer length of SAT DNA sequence range from 150 to 400 BP in the majority of plants and animals. SAT DNA sequences are located at heterochromatic regions, which are found mostly in centromeric and subtelomeric regions in the chromosome, but also at intercalary positions. A satellite DNA are chiefly non-coding. It does not code for a particular protein. Tandemly repeating DNA is also called a satellite DNA because DNA fragments containing tandemly repeating sequences from satellite bands and genomic DNA are fractioned by density gradient centrifugation. For example, when broken into fragments, human DNA contains a main band like this, and the main band contains DNA fragments made up of mostly single copy sequences with GC composition close to 40.3%. The average value for the human genome, the satellite bonds contain fragments of repetitive DNA and hence have GC contents and band densities that are atypical of the genome as a whole. So satellite DNA may be generated by some of the chromosome level process, namely unequal crossing over during meiosis and unequal sister chromatid exchange at mitosis. They are involved in the formation of heterochromatin. So periodic AT distribution leads to the curvature of DNA, and satellite DNA with its AT-rich regions is considered to be important for packing of DNA in heterochromatin region. The length of a satellite DNA takes from 1 to several base pairs. For instance, in humans, and alphoid DNA is a satellite DNA found in all chromosomes and consists of 70 base pairs. Another is the beta satellite DNA found in the centromeres of chromosome 1, 9, 13, 14, 15, like that. It consists of 68 base pairs. Satellite DNA classification. It represents a class of repetitive elements consisting of a cluster of short repeated sequences, and it is divided into several subcategories according to the size of the individual repeat. Mini satellites. Mini satellites vary in repeat size from 9 to 100 base pairs, usually about 15 base pairs, and is usually found in clusters of 10 to 100 repeats located in euchromatic subtelomeric regions or dispersed in other areas of the genome. It is also called variable number tandem repeats, VNTRs. Telomeric DNA, which in humans comprise hundreds of copies of the motif, is an example of mini satellite. Then microsatellite. Microsatellite consists of a very small sequence repeated 10 to 100 times per cluster and located in various places in the euchromatic portion of the genome. It is also called a shorter tandem repeats. Microsatellites are thought to have originated by polymerase slippage during DNA replication. The commonest type of DNA, human microsatellite, are dinucleotide repeat, which approximately this much amount of copies in the genome as a whole, about half of this being repeats of motif CA. What is the application of this? The short sequences of DNA structures in DNA, satellite DNA vary from individual to individual. These variations in the length and sequence of satellite DNA are unique to each individual. This uniqueness can be exploited to identify each individual based on their DNA map. This technique is called DNA fingerprinting, and it is used to identify criminals, perform paternity tests, and diagnose genetic disorders. Selfish DNA. Selfish genetic elements. Selfish genes, ultra-selfish genes, selfish DNA, parasitic DNA, and genomic outlaws are genetic segments that can enhance their own transmission at the expense of other genes in the genome, even if it has no positive or a negative effect on organismal fitness. Selfish DNA is a concept that was proposed by British evolutionary biologist Richard Hawkins, and selfish DNA refers to those sequences of DNA which in their purest form has two distinct properties. DNA sequence spreads by formal additional copies of itself within the genome. It takes no specific regulation to the reproductive success of the host organism. One well-known example of selfish DNA is transposable element, also known as jumping genes. These are DNA sequences that can move around within a genome, often by copying themselves and inserting the duplicates into different locations. Transposable elements make a significant portion of many genomes, including humans, while some transposable elements have been co-opted by the host organism for beneficial functions. Many others are considered selfish because they can disrupt important genes or regulatory regions, leading to harmful effects on the organism's fitness. Understanding the behavior of selfish DNA has important implications for various fields, including genetics, evolutionary biology, and medicine. The study of transposable elements and their impact on genomes can shed light on the genetic diseases, evolutionary history, and genome regulation. Furthermore, harnessing the power of selfish genes biology, genetic elements may have applications in genetic engineering and synthetic biology. Transposable elements, the chromosome, selfish DNA. Over many generations, rare intact element, deletion, mutation. Conclusion. Scientists have found that some junk or non-coding DNA are essential to optimum cell functioning, particularly in regulating gene activity. Recent research has identified non-coding DNA sequences that are found in nearly all plants and appear to have roles in basic processes such as tissue and organ development, response to hormones, and regulation of gene expression. Junk DNA may provide a reservoir of sequences from which potential advantageous new genes can emerge. In this way, it may be important in genetic basis for evolution. Satellite DNA is composed of a very large array of randomly repeating DNA. That means a single sequence of DNA is repeated many times over side by side. They are non-coding DNA in nature. Satellite DNA is a major ingredient of functional centromere and builds the main structure of chromosome, heterochromatin. They hold highly repetitive DNA. The major characteristics of satellite DNA is its uniqueness or the variation between individuals. For these features, it is used in DNA fingerprinting. Selfish genetic elements have now been described in most groups of organisms, and they demonstrate a remarkable diversity in the ways by which they promote their own transmission. Although long dismissed as genetic curiosities with little relevance for evolution, they are now recognized to affect a wide swath of biological process ranging from genome size and architecture to speciation.